Everyone, this is a next video lecture on the chapter sexual reproduction in flowering plants. Students, in this chapter we mainly already studied about the structure of flower and the important reproductive organs in case of flower, both male and female reproductive organs. In case of male we studied about the stamen, and in case of female we studied about the pistil or carpel, or you can say it as a gynosia. And in the last video lecture we mainly studied about the process of megasporogenesis, how the megaspore is going to be formed, which is a which is not female, which is a oval. Okay, this megaspore is commonly we call it as the oval, and the process of formation of megaspore we call it as the megasporogenesis. This completely we studied with the help of this important diagram. Okay, this is the diagrammatic representation that's showing uh, megasporogenesis. Okay, here the nucellus is there. From the one of the cell from the nucellus is undergoing division, mitotic division. It will grow and it will undergo the mitotic division, and at the finally they are going to form four megaspores, and that condition we call it as a megaspore tetrad. Among four, three of them are going to be disappear, and only one is going to be develop into embryo sac or female gametophyte. This is the thing what we studied in the last video lecture. And moving to the next one, moving to the next important concept, that is the development of female gametophyte. This one, how the female gametophyte is going to be developed from here to here, how it is going to be developed, because there is arrangement of cells and everything we know. Okay, but in case of megasporogenesis, what will happen? Only one megaspore left, only one megaspore is left, and there is different types of cells they are going to be arranged, nucleus, everything they are going to be formed. If you observe the matured one, it is completely different from the megaspore what we found after the meiosis. So lot of cells are going to be formed here, lot of nucleus they are going to be divided here. How they are going to be divided and how they are going to be arranged, that we will study with the help of this diagram. Okay, and in the slide we mainly given these all things. These are the important slides, the important points what we have here, but with the help of that diagram I will try to convey you the entire concept. Okay, first one, what we will do here, this is the megaspore. This is the megaspore, which contains, in the beginning, which it contains only one nucleus. It contains only one nucleus, not two. Okay, for example, after completing degenerating one, there is only one nucleus, one cell is there, and inside that only one nucleus is there. Okay, and what will happen, this megaspore undergo karyokinesis. Students, please remember, it will undergo karyokinesis, not cytokinesis. It will undergo karyokinesis. Karyokinesis is nothing but division of nucleus. It is nothing but division of nucleus, not the cell. Okay, this megaspore will undergo karyokinesis. That is very important, please remember, karyokinesis. This is the megaspore what formed in the beginning. It will undergo karyokinesis. Okay, it means these this single nucleus, okay, whatever the cell formed here, this single nucleus will undergo division. What division? It will undergo the division mitosis, not meiosis. It will undergo the division mitosis. What will happen in the mitosis? One cell become two, one become two, one cell become two. Here what will happen? One nucleus become two nucleus. It will undergo mitosis. Okay, it will undergo mitosis. And what is going to be formed here? In a single megaspore there is a presence of two nucleus, and this stage is two nucleated megaspore. present here. And again what will happen, just two nucleated megaspore, again the nucleus will undergo division, it will undergo further mitotic division, and there is a formation of four nucleus. Okay, one for one nucleus, another for another nucleus. Totally there is a four. And these nucleus will move to opposite pole. This is a one pole and this is another pole. They will move to opposite poles. They are going to be moved to opposite poles. Okay. And after that again what will undergo nuclear division. So the second stage of four nucleated megaspore is found here. First division, two nucleated megaspore. Second division, four nucleated megaspore. This is the first division, second division. Now in next division, 4 becomes 8. 8 nucleus they are going to be forming. 4 become here 8. So total 8 nucleus they are going to be forming. This is important. This is important. Please remember single megaspore. Single megaspore which contain giant process, that is division of the nucleus, and this karyokinesis leads to the formation of 8 nuclei. So total 8 nuclei they are going to be forming. In the first division 2 nuclei, that is 2 nucleated stage. In the next division 4 nuclei, that is 4 nucleated stage. In the next division they become 8 nucleated stage they are going to be forming. This is important. This is how the cells are going to be divided. Okay, this is the importance of formation of the. Okay, 8 nuclei they are going to be formed the final result. But one thing students, please remember, after the division, first division and second division, there is no immediate development of the cell wall. Cell wall is not going to be laid down immediately. It will wait for a time up to their complete division. Okay, they will wait for a few minutes, after the few minutes just after completing the cell division, sorry not cell division, nuclear division, that nuclear division, after that there is a development of the cell wall. It means karyokinesis is not immediately cytokinesis is not immediately done by karyokinesis. They will wait for a time, and after that what will happen, we'll study about the arrangement of the cells, how they are arranged and where they are arranged, that we will study here. Here there is a chalajal end, here there is a microfilar end. Totally 8 cells are, totally 8 nuclei are formed here, and immediately the cell wall is going to be start development. Okay, first students, there is a presence of 8 nuclei. Okay, 8 nuclei. And how the cells will arrange, first I will tell you the name of the cells. 3 antipodal cells, 2 synergid cells, 1 egg cell, and 2 polar nuclei. Okay, 3 antipodal cells, 2 polar nuclei, 2 synergid cells, and 1 egg cell is going to be formed here. But the cells, how many cells are here? There is a 7 cells, not 8 cells. 8 nuclei are there, but 7 cells are there. Not 8 cells. 7 cells are there. Please remember. And before that, I wanted to discuss about some important thing, that is ploidy of the nucellus. What is the ploidy of nucellus? It is 2n. Please remember. Ploidy of the nucellus is 2n. What is meant by ploidy? That number of sets of chromosomes. Okay, it is 2n in case of nucellus. In case of microspore mother cell, that is pollen mother cell, it is also 2n. Please remember. Then megaspore mother cell, it is also 2n. Please remember. But megaspore is n, haploid. It's not diploid, it is haploid, because megaspore mother cells will undergo the division, and that will leads final formation of the haploid. Okay, please remember the number of chromosomes. Okay, there is a possibility of asking 1 marks question. Okay, name the ploidy of megaspore mother cell, or name the ploidy of nucellus. They will ask the question like that for 1 marks. Okay, please remember. Okay, this is the structure and how they are arranged, how they are going to be arranged. First, here 8 sorry here first there is a chalajaland, and 3 cells are going to be formed in the chalajaland, and those we call it as the antipodal cells. And what is the function of antipodal cells? Antipodal cells will nourish the embryo sac. Please remember, antipodal cells will nourish the embryo sac. It is going to be nourish the embryo sac. Antipodal cell. Okay, 3 cells are there. 1, 2, 3. 3 cells are there. How many remained? The remaining is 4 cells. And 2 synergid cells. Here you can find 2 synergid cells. This is 1, this is 2. 2 synergid cells. And along with the synergid cells, there is a egg cell. Along with the synergid cell, there is a egg cell. Okay, just wait, I will clarify it. Just wait. Here is a diagram. Okay, this is clear on. You can see here. Antipodal cells, this is 1, 2, and 3. And one here synergid cell you can find here. And one egg cell. Along with the synergid cell, we will find the egg cell. Okay, and synergid cells also contain some cellular structures we can find here. Okay, cellular structure or cellular thickenings we can find here. Here they are present exactly, and these we call it as a piliform apparatus. Please remember, piliform apparatus. In synergid cells, there is a presence of 2 cellular structures. Okay, and these cellular thickenings we call it as a piliform apparatus. What is the function of piliform apparatus? These will help in the guidance of pollen tube for the transfer of the gametes. Okay, it mainly guide the pollen tube to release the gametes into the synergid cell. It will give the guidance or direction. The same is that of the synergid cells. The same function we can say for the synergid cells. They will guide the pollen tube for the transfer of gametes. Okay, this is about the synergid cells. And coming to the next, that is egg cell. Okay, just along with the synergid cell, we can find the egg cell, which is the main important for the fusion of the gamete. Okay, this is our main target. Here the must and should there is a fusion of the gamete can takes place. Okay, in the egg cell, and together the egg cell and the synergid cell together we call it as the egg apparatus. In the micropolar end we can find the egg apparatus, and after the egg we'll find another one type of cell. This cell, this complete cell. Okay, this one cell, it contains 2 polar nuclei. It contains 2 polar nuclei. During double fertilization we will study about all these things. Okay, how the embryo is going to be develop and everything we'll study here in the later classes. Okay, in the later classes we will study about all these things. Okay, first, but here you can find polar nuclei in the middle of the cell. In the middle we will find the polar nuclei. This one, 2 nuclei are there. 2 nuclei, those we call it as the polar nuclei. How many cells are over here? 3 antipodal cells, 2 synergid cells, 3 antipodal cells, 2 synergid cells, 1 egg cell, and 2 polar nuclei. So sorry, not polar 2 cell, one middle central cell. One middle central cell. How many cells are over now? 3 plus 2, 5, 6, 7. Total 7 cells. But nuclei are 8 here. 1 nuclei, 2, 3, 4, 5, 6, 7, 8. Total 8 nuclei, and 7 cells are present in case of the female gametophyte or embryo sac. Okay, embryo sac. This is the thing. Like this, there is an arrangement. Please remember the function of antipodal cell. It will mainly nourish the embryo sac. Then synergid cell will guide the pollen tube for the fertilization. Please remember, these are important. Okay, and in the middle we can find here polar nuclei. Okay, in the double fertilization we'll study about that, how they are going to be fertilized and everything. But here they given only the structure of this female gametophyte. And students, please remember, this is very important. The last year question paper only in March 2020, they asked the question, explain the structure of female gametophyte. For that you have to draw the diagram and you have to explain all these cells, where they are present and then their functions. Okay, for that please remember these things. Okay, I think so you all of you understood this concept. Okay, I think so all of you understood this entire concept. If you have any doubt you can ask. And another one thing, students, please remember here, here monosporic development occurs here. Monosporic means only the embryo sac is going to be developed from a single megaspore. From a single megaspore, one embryo sac is going to be developed. So this condition we call it as a monosporic. Okay, this condition we call it as a monosporic. Okay, this is the explanation, students. Please go through the video and read your NCRT textbook and all. I think so all the points I have covered in this video lecture. I think so from the NCRT, whatever the NCRT given, those all points I have covered here. And if you have any doubt you can ask. And these are all the points here, female gametophyte, these are all the points what I was collected. These are all the points point wise, and whatever the things I was explained now, the same thing is present in case of in this slide. Okay, the same thing present here. And moving to the next one, the same points here, the same whatever the things I was explained here, synergy cells, one egg cell, everything is present here. And please go through the video and read repeated times. And if you have any doubt you can ask the question through WhatsApp, and I will try to clarify your doubts and then I will try to convey your concept. Okay, thank you. And in the next video lecture we will begin with pollination. Okay, thank you, students.